Are the slides all loaded? It's a miracle because they did not have them 20 seconds ago. So thank you very much to our ACE technical team. So yeah, it's great to see everybody today. I'm very excited about the workshop. I want to thank the organizers, Martin and Alistair, for the time and effort, and all the staff for planning this, and I'm happy to kind of help kick it off. This was the title I was given, and I thought it was long enough that I wouldn't change it. Transforming our ability to generate, analyze, integrate, and share information. Really, this is about the opportunities for informatics and data science in regulatory science. As many of you know, that's what I do when I'm not chairing the drug forum. And so I want to tell you about some of the stuff we've been doing, but really emphasize the impact that I think the data sciences and informatics can have in regulatory science. So I'm just going to very briefly tell you, and many of you know this already, that there's a Center of Excellence program that the FDA sponsors called the Centers of Excellence for Regulatory Science and Innovation. And I am co-PI with my colleague Kathy Giacomini of the UCSF Stanford CIRSE, who has 3 goals: to create a West Coast presence, and we have a visiting program, and I won't talk about that; to promote research and education in innovative regulatory sciences, I will talk a little bit about that; and to facilitate multi-sector interactions in regulatory sciences, which is incredibly important and fun. On the research and education, we decided early on that our CIRSE would be focusing in 2 areas: next-generation biomarkers and data science. And obviously, those 2 are very closely interlinked, and I'll talk about the next-generation biomarkers a little bit at the end. And I'll talk about data science right now. So this is the team, especially my co-PI and really the founder of this idea of writing this proposal was Kathy Giacomini at UCSF, and we work very closely and have a great time. And before I forget, I want to give a plug for our YouTube channel. So and this is fun, but it's also important. One of the challenges, especially at Stanford, is we don't have a legacy of regulatory science activity on the campus, or at least regulatory science activity that people recognized as regulatory science. So we have a training and education opportunity for postdocs and graduate students so that they see what this field is, what people who do it think they're doing, and how others can get involved. And so when we do host visitors from FDA and from industry talking about regulatory science, we routinely capture them on video. And then if they're FDA employees especially, who are serving the country and who are national federal employees, it's for everybody. And so we actually put it up on this YouTube channel where we are now starting to have a pretty reasonable curriculum of regulatory science in the words of regulatory scientists. And we count hits and we do all those kinds of things. But this is critical because it gives me an easy answer to the question from a young person: how do I find out what regulatory science is? I'm a mechanical engineer, or I'm a biomedical informatician. I've heard about this thing, regulatory science, and so we have courses, we have workshops, and for those who don't want to do anything except between the hours of 12 a.m. and 6 a.m., they can watch these at their apartment. So the story, the very short story I want to tell is, when we first started the CIRSE, we decided it was very important not to have it be curiosity-driven research, which is what we normally do at Stanford and everywhere else. But we wanted to be mission-driven for the FDA mission. And so we actually went out to colleagues at the FDA, and this was with the help of Frank Weichhold, York Tomita, and others who are running the CIRSE program and who are here today. We kind of tried to beat the bushes and look for problems where we could help. And we had kind of declared our interest and expertise, but we very much wanted to hear from, in this case, FDA scientists about collaborative projects that they thought would be meritorious and could take advantage of the skills and talents of our faculty locally. And we got 22 or maybe even more proposals, and in the last 2 years we've funded either pilot or not so pilot projects in these 13 areas. And you can see that it was from a diversity of centers, excuse me. We haven't gotten all the centers, but we're working on that. We want to make sure we kind of fully engage all aspects of the FDA. But really what I want to focus on is these ones in red are ones that are squarely have large data science components. So I just want to describe those in a couple of sentences. And the theme here is that the FDA and us recognize that there are places where informatics and data science can make impact right away and This is not a random sampling, so I wouldn't say that 5 or 6 out of 13 is the baseline rate of data science problems, because they knew about our interests and our expertise. But nonetheless, this is, I think, a nice sampling of the kinds of things that you see. So the first one is improving the efficiency and rigor of pharmacovigilance. So this is a project being done by one of my students with Bob Ball at the FDA, looking at the FAERS incoming reports. It turns out that not all FAERS, this is the FDA Adverse Event Reporting System, not all of those spontaneous reports are of equal value. Some have very high value, and the reviewers are very appreciative because it gives them information to perhaps follow a lead, and others are, for a variety of reasons, not very useful. And the question is, can automatic machine learning triage those reports and put in front of the reviewers the ones that are deemed to be likely high value? So we've been having this great project for now a few months, 6 months, where FDA reviewers actually gave us a set of 500 reports that they graded on a scale of 1 to 7, 1 being the most valuable, 7 being dubious value. And then we are using some kind of modern machine learning techniques to see how well we can reproduce that on the training set. And then they've held out some that we haven't even seen yet. So when we say, okay, we think we have a good classifier, they're going to give us those, and we'll get kind of the final performance on a set of gold standard. So a lot of fun figuring out what are the signals in these reports, and something that, if it's successful, could accelerate the ability of the FDA to go through these spontaneous reports. Improving the diagnostic accuracy of adverse event signals. This is a very simple idea. It's headed up by Nigam Shah, my colleague at Stanford, with FDA collaborators again. The idea here is you might see a signal in FAERS, the database I was just discussing, but we also have electronic medical records. We have them at Stanford. We also have them as part of the Mini Sentinel and Sentinel projects. Can we combine the evidence from the electronic medical record with the signals that are being seen in the adverse event systems to get even more confidence that something is true, or even less confidence that it's true, based on the combination of the data sources? So this, of course, relies on electronic medical record infrastructure, but is incredibly exciting as a way to, what we call, do data fusion to improve our ability to triage signals. E-source pathology checklist. This is up at UCSF, Laura Esserman, our colleague. This is a electronic medical record infrastructure that will actually prod physicians to record important pieces of information that are critical either for a clinical trial or for quality insurance or quality outcomes measurement. It's that all information is not equal, and if you have a system in the background watching the electronic medical record and there's an important piece of information that you really need for a trial, for care, for any reason, you can, in a judicious way, kind of ping the physician and say, you need to fill this out, it's important. And it's the question of how to do that both socially and technically, and not become hated. And then on the bottom here, we have a project with the Office of Generic Drugs and about excipients, and that might sound, well, wait a minute, that sounds like a lab project, and in fact there is a laboratory component, but a large component of this is to understand the biological activity of excipients, especially as they may interact with either the active compound in that capsule or other compounds that the patient might be taking. And a large fraction of that project, which is being done by Brian Scheuket, colleague up at UCSF, is to use computational chemistry methods to look at the chemical structures of the excipients and see if you can predict unanticipated interactions either with cytochromes or with transporters or with other molecules that might make these otherwise apparently inert excipients not quite so inert in the context of a particular drug. So we're very excited about that project, which is just getting off the ground. And finally, in a suite of projects in my lab, we are looking at, one I would like to highlight is knowledge management. The FDA has huge treasures of unstructured data, text, often PDF, sometimes ASCII text. And in a sense, like many organizations, FDA might not fully know what it knows because of the information that is latent in these collections. A colleague of mine at Stanford, Chris Ray, just won a MacArthur Award for his work in building computer systems to wrestle such a corpora of data to the ground and get them organized. And so we are working with Chris to use his system, which is called Deep Dive, on the public assets of the FDA first to see what we can figure out about publicly available information, linking it together and making new inferences. But with success, we will be able to translate this into the internal proprietary assets of the FDA, maybe not us, somebody who has the right clearance, so to speak, and then perhaps create a knowledge management infrastructure that could really transform the ability to find out what the FDA knows. And we're also looking at our participation in the Precision FDA effort, which is the informatics component of the precision medicine work being done at the FDA as part of the President's precision medicine. So really, the next two slides are really my take-home message. They're in the middle of the talk, but if I were to tell you 10 key data science themes, I just want to kind of, based on the first year and a half of our CIRSE, lots of confirmations with lots of conversations with colleagues, both at FDA and at the European agencies and at other regulatory science interested bodies. These are the things that come up all the time. So I just mentioned knowledge management, bringing large unstructured text into a system where it can be queried and used for knowledge management. Integrating information across all phases of drug evaluations, including post-marketing, in order to detect efficacy and safety signals more sensitively, such as the project I talked about with the EMR and the FAERS data. I told you about our automated triage and prioritization. We're doing this for FAERS, but you could do it for the device adverse events reports. You could do it for the vaccine adverse events reports. So we think of this as just the first step. Integration of spontaneous data reports with EMR, that's the one I meant with NIGM. Computational infrastructure and statistical models for approval of next generation biomarkers, which I'll talk about a little bit more, and I think you're going to hear more about in this meeting. Standards for mobile health software quality. You can't just fix a bug in a piece of FDA-regulated software, even if it's on an iPhone. You need to fix the bug and then make sure you haven't broken the whole system. And there are principles of software engineering there that are relevant. Validated electronic infrastructure for clinical trials, not unrelated to that eSource pathology project that I mentioned. Using social media for both surveillance, vigilance, and for indirectly patient-reported outcomes, which I'll come to in a second. Systems pharmacology. Can we get computational models? Darrell Abernathy ran a great meeting about a month ago in this topic, and there's great interest in creating computational models of biology and biological pathways that can be used to anticipate and understand drug response data. And finally, an electronic infrastructure for patient-recorded outcomes and for elicitation of patient preferences. I know this is a focus at CDRH these days, and very important, and it's all going to happen on an iPhone probably. So thinking about those issues, very important. So I just want to finish up, and I think I have time. I think I'm okay on time. You all know that, so everybody I know who does genomics or bioinformatics, this is their new favorite photograph, right? I mean, the president is pointing at a DNA double helix. That doesn't happen every day. So, and he announced the Precision Medicine Initiative, and part of that is the Precision FDA Initiative. And I just want to tell you a little bit about that as an example. I think Dr. Kashoot might be here or will be here. Taha will be here. And we've been working very closely. I care about this because we've been building a database on pharmacogenomics for the last 15 years, and we're very interested in getting this out into implementation. So on September 25th, we had this great forum with CDRH scientists, NIST scientists, including Dr. Salat, who's here today. And we talked about this Precision FDA, which is a collaboration between the FDA, vendors, contractors, and grantees to build an open source cloud infrastructure for supporting regulatory science for next generation sequencing. This is not the platform that will be used for regulatory decision making. This is the platform for the regulatory science, although obviously it will inform regulatory decision making and how it's done. So focus on software. The idea is to share genomes, share pipeline software, share evaluation, and to get an initial release of this playground for the community to look at in mid-December. You know, this is a very important, next generation sequencing is a great example of a next generation biomarker. And they have funky features that require some attention. They make a lot of measurements in order to be cost effective. So it's not one measurement for one decision. It's one measurement, millions of decisions, and that's stressful. They may discover uncharacterized and unexpected variation that need to be understood and triaged by the clinician. Full validation is difficult to imagine. There are 3 billion bases in the genome. It's going to be hard to validate that we get every base right every time, and yet we want to move forward somehow with a framework that's flexible and that can deal with that. We don't always have a specific intended use. Sometimes you can think of it more as a screening test, and the rules for screening tests and for focused diagnostics are very different, and yet you're kind of doing both at the same time. That's also stressful. Each platform has different performance characteristics. A computational pipeline is mandatory for the readout, and therefore informatics is important, and human inspection is often still required. So this is what we discussed at the workshop. We came up with a roadmap, which I don't have time to go over, but it basically is the to-do list, and you might hear a little bit more about it from Mark later today. So in conclusion, we have this exciting set of regulatory science challenges for data science and informatics. We've been very gratified by the enthusiasm of our FDA collaborators and external partners to tackle these challenges. And as I said, one of our main goals is to do the basic education to well-trained scientists so that they get excited about regulatory science and add their skills to the mix. So I'll stop there and thank you for your attention.